Welcome to the deep dive. Today we are really getting into the weeds of modern medical science, molecular diagnostics. We're talking about the incredibly powerful tools scientists have now to read our genetic code, and not just read it, but understand it and use it to diagnose conditions. Our mission for you today is pretty straightforward. Take that stack of materials you've got, the lab techniques, the papers, and boil it down. We want you to walk away with a really solid grasp of three key techniques: PCR, Sanger sequencing, and the big one, next generation sequencing, or NGS.
Yeah, and it's crucial to understand why these matter. These aren't just abstract lab methods. They're literally the tools used every day to figure out what's going on genetically.
Yeah.
Think about conditions like Noonan syndrome. These techniques let us find the cause, like understanding how certain pathways get hyperactivated. So we'll cover the basics, the workflows, and, importantly, what kinds of genetic changes each method can actually detect.
Okay, sounds good. Let's dive in. Where better to start than the molecular copy machine itself?
Mhm.
PCR, polymerase chain reaction, invented back in '83 by Carey Mullis.
The concept is actually surprisingly simple, isn't it? Just make lots and lots of copies of one specific bit of DNA in vitro.
Deceptively simple, maybe, but totally revolutionary. Because making billions of copies from potentially just one starting molecule, that underpins almost everything else we do in molecular biology.
Yeah.
It's the essential first step so often.
Right, it lets you see something that was basically invisible before.
Exactly. And it all happens in this machine, the thermal cycler, through repeated temperature changes, usually, 25 to 40 cycles?
Something like that, yeah.
And each cycle has three key steps. First step is denaturation.
Getting things hot.
Really hot, about 95 degrees Celsius. That heat breaks the hydrogen bonds holding the two DNA strands together. Pop. Separate.
Okay, step 1, separate strands. Step 2, annealing. We cool it down quite a bit, maybe 50 to 65 degrees C. This lets these short, custom-made DNA pieces called primers find and stick to the specific spot on the DNA that we actually want to copy.
So the primers mark the target.
They define the target sequence. And then step 3 is extension, usually around 72 degrees Celsius. This is where the enzyme, the DNA polymerase, gets to work.
Ah, the polymerase. Now, wait a second. You just heated everything to 95 degrees. Wouldn't that normally destroy an enzyme, a protein?
Absolutely. Most enzymes would just fall apart. That was the big hurdle initially. The breakthrough came from finding an enzyme that could handle the heat.
From that bacteria in the hot springs.
Exactly. Thermus aquaticus, living happily in places like Yellowstone hot springs. Its DNA polymerase, Taq polymerase, is thermostable. It shrugs off that 95-degree heat.
Incredible.
So at 72 degrees, Taq latches onto the primer and starts adding nucleotides, building a new DNA strand complementary to the template.
And because you repeat this cycle, denature, anneal, extend.
Over and over.
The amount of target DNA just explodes. It's exponential growth, right? 2 copies become 4, 4 become 8.
Precisely. After about 30 cycles, you've gone from maybe one starting molecule to over 1 billion copies. It's an astonishing amplification factor.
Which is powerful, but I guess it also brings risks.
Oh, definitely. You need the right ingredients. Your template DNA, those specific primers, the nucleotide building blocks, DNTPs, and the Taq enzyme. But the biggest risk by far is contamination.
Right, because if even a tiny speck of unwanted DNA gets in.
It gets amplified billions of times too. Instant false positive. That's why molecular labs are almost paranoid about clean workflows, separating pre-PCR and post-PCR areas. Primer design is also critical. They need to be specific to only your target.
Okay, so PCR gives us the quantity. But we need the quality, the actual sequence, A's, T's, G's, C's. That takes us to the classic method, Sanger sequencing.
Yep, Fred Sanger's Nobel-winning technique from 1977. It's built on a really clever idea called chain termination.
Chain termination. Explain that.
Okay, so in the sequencing reaction, you have the usual stuff. Template DNA, primer, polymerase, and the normal DNA building blocks, the DNTPs. But you also add a small amount of special nucleotides, dideoxynucleotides, or DDNTPs.
Deoxy. So they're missing something.
Exactly. Normal DNTPs have a little chemical hook, an iso-H group, on their sugar component. That's where the next nucleotide attaches. Dideoxynucleotides are missing that hook.
Ah, so if the polymerase adds one of those.
The chain stops, dead end. Synthesis terminates right there. Since the incorporation of a DDNTP is random, you end up creating a whole collection of DNA fragments, all starting at the primer, but ending at every possible base position where a DDNTP got added.
And originally that meant 4 separate reactions, one for each DDNTP base.
Yes, but the modern way is much slicker. Now you do it all in one tube. Each of the four DNTTs, A, T, G, and C, has a different colored fluorescent tag attached.
Okay, so you get a mix of different length fragments, each ending with a specific colored base. How do you do that?
You separate these fragments by size using a technique called capillary electrophoresis, basically pushing them through a very thin tube filled with the gel matrix. Shorter fragments move faster.
Right.
As the fragments zip past a laser detector, sorted by size from shortest to longest, the laser makes the fluorescent tag light up. The detector reads the color.
And the sequence of colors tells you the sequence of bases.
Precisely. You get this readout, a chromatogram showing the peaks of color. It's incredibly reliable, the gold standard for accuracy, really.
But it has limits, right? You mentioned fragment length.
Yeah, that's the main constraint. Reliably separating and reading fragments much longer than about 800 base pairs gets tricky with Sanger. It's perfect for single genes or specific regions, but not for huge amounts of sequence.
Which brings us neatly to why we need more capacity. Let's make this concrete. Talk about Olivia's case. Okay, so Olivia, she's 9 now. Her story started before birth, actually, with an ultrasound showing a cystic hygroma. After birth, exams showed some distinctive features. Downward slanting eyes, low-set ears, wide-spaced nipples, and critically, heart problems.
An atrial septal defect and pulmonary valve stenosis.
Yeah, serious congenital heart disease.
And that whole picture, the facial features, the heart defects, strongly points towards Noonan syndrome.
Okay, what should we know about Noonan?
Well, it's relatively common for a genetic syndrome, maybe 1 in 1,000 to 1 in 2,500 births. Usually inherited as autosomal dominant, meaning just one copy of the altered gene is enough. And in Olivia's case, the specific variant mentioned in the sources is in the PTPN11 gene.
A specific spelling mistake in the code.
Exactly. The notation is NM00028345, PTPN11.C.166AG. That means at position 166 in the PTPN11 gene's coding sequence, an adenine A has been changed to a guanine G. This results in an amino acid change in the protein. Isoleucine becomes valine at position 56, PIL56-FAL.
And why does that one change cause Noonan syndrome?
It comes down to cell signaling. PTPN11 and other genes often involved like SOS1 or RAF1 are key players in a pathway called the RAS-MAT pathway. Think of it like a major communication highway inside cells controlling growth and development.
Okay.
These specific Noonan-causing variants are usually gain-of-function. They basically jam the accelerator pedal on this pathway. It becomes hyperactivated, constantly on, leading to the kind of unregulated growth and differentiation problems you see in Noonan syndrome.
Right, so if you suspect Noonan, you need to check PTPN11, maybe SOS1, RAF1. How many genes are we talking about? PTPN11 alone has 16 exons. BRAF has 20.
It adds up quickly. You could be looking at dozens of genes, potentially hundreds of exons in total.
And trying to check all of that using Sanger sequencing 800 base pairs at a time.
Forget it. It would take forever, cost a fortune, just completely impractical for this kind of multi-gene question.
Enter the heavyweight, next generation sequencing, NGS.
This is where the paradigm shifts. If Sanger reads one piece of DNA meticulously, NGS reads millions or even billions of pieces simultaneously. It's massively parallel.
Massively parallel. Okay, break that down. How does it work? Doesn't sound simple.
The core idea is different. First, you take the patient's DNA and smash it into millions of tiny fragments, randomly.
Okay, chop it all up.
Then you stick special DNA sequences, called adapters, onto the ends of every single fragment. This creates what's called a sequencing library.
Library prep. Got it.
Next, you flow this library over a special glass slide, the flow cell. The surface of this flow cell is coated with short DNA sequences that match the adapters, so the fragments stick down.
They hybridize to the surface.
Right, and then comes a clever amplification step right there on the slide, often called bridge amplification. A fragment attaches, bends over to form a bridge to another spot, and gets copied. This happens over and over, creating a tiny, dense cluster of hundreds of identical copies of that original fragment. You end up with millions of these distinct clusters all over the flow cell.
Wow. So millions of little reaction zones, each with copies of one original DNA piece.
Exactly. Now you're ready to sequence them all at once. And this is where NGS differs chemically from Sanger too. It uses reversible terminator DNTPs.
Sanger's terminators were permanent stops.
Precisely. With NGS, you flood the flow cell with all 4 fluorescently labeled reversible terminators and polymerase. In each cluster, the polymerase adds one correct base. A laser scans the flow cell, records the color emitted by each cluster, identifying the base that was just added.
Okay, read the base.
Then, crucially, a chemical step removes both the fluorescent tag and the blocking group that made it a terminator. This regenerates the 3OH hook.
So the chain can be extended again in the next cycle.
You got it. Add the terminators again, image the colors, chemically cleave, repeat hundreds of times. Each cycle reads the next base for every single cluster on the flow cell in parallel.
Mind-boggling scale. But that must generate just mountains of data, all those color signals from millions of clusters over hundreds of cycles.
Absolutely enormous amounts of raw data. The challenge shifts from the wet lab to the computer bioinformatics.
So what's the data analysis pipeline look like? How do you make sense of it?
First, the imaging data gets converted into actual sequences, base calls, ATGC, along with a quality score for each base. These are your raw read files, usually in FASTQ format.
Okay, FASTQ equals raw reads plus quality.
Then you need to figure out where each of those millions of short reads actually came from in the human genome. That's alignment or mapping. Software compares each read to the reference genome sequence. The results are stored in BAM or CRAM files.
BAM files map the reads to the genome, like putting the shredded documents back together.
A good analogy. The final critical step is variant calling. The software compares the patient's map sequence in the BAM file to the reference genome and identifies all the differences, single base changes, small insertions, deletions. These variants are listed in a VCF file, variant call format.
VCF. That's the file the geneticist looks at to find the potential disease-causing change, like Olivia's PTPN11 variant.
Exactly. That's the output that links back to the clinic.
Now, when a lab decides to use NGS, they have choices, right? Different flavors of NGS.
Right. There are three main approaches. You could go for whole genome sequencing, WGS. That aims to sequence essentially everything, all 3 billion base pairs, coding and non-coding regions. Massive data set.
A whole enchilada.
Then there's whole exome sequencing, WES. This targets just the protein coding regions, the exons. That's only about 1.5% of the genome, but it's where most known disease-causing mutations are found. Much less data than WGS, more focus.
Okay, just the exons.
And the third option, often very practical, is panel sequencing. Here, you only capture and sequence a specific, pre-selected set of genes known to be associated with a particular condition. For Noonan syndrome, you'd have a panel covering PTPN11, SOS1, RAF1, and the other known Noonan genes.
Ah, so super targeted.
Highly targeted. Reduces cost, simplifies the data analysis hugely because you're looking at far less sequence, and importantly, it minimizes the chance of finding incidental findings variants in unrelated genes that might cause anxiety or require further investigation.
So for Olivia, suspecting Noonan syndrome, where we have a good list of known causative genes, a panel, or maybe WES, makes the most sense clinically.
Generally, yes. It's often the most efficient path to a diagnosis in that situation. You get deep coverage of the relevant genes relatively quickly and cost-effectively. WGS might be used if the panel or exome comes back negative, suggesting a cause might lie outside the usual suspects, maybe in a non-coding region.
It really is about picking the right tool for the specific question you're asking.
Absolutely. You wouldn't use a sledgehammer to crack a nut, right? You wouldn't necessarily jump to sequencing the entire genome if a focused panel can give you the answer faster and cheaper for a condition like Noonan. It's about balancing diagnostic yield, cost, turnaround time, and the complexity of interpretation.
Okay, let's pull this all together then. We've journeyed from PCR, the essential amplifier,
Making the invisible visible.
to Sanger sequencing, the precise classic reader for shorter stretches.
Gold standard accuracy, but limited scale.
And finally, to the powerhouse of NGS, enabling that massive parallel sequencing needed for complex genetic questions like diagnosing Noonan syndrome by looking at many genes at once.
Yeah, understanding how these tools differ, how they evolved, and where each fits in is key. It really boils down to matching the technology to the clinical or research need.
Perfect. So that leaves us with our final thought for you to chew on. We've seen how targeted approaches like panel sequencing and WES are often the practical choice today. But
Yeah.
we also know that whole genome sequencing captures everything, including all those non-coding bits, the regulatory regions, the stuff we, frankly, still don't fully understand the function of. As WGS gets cheaper and faster, what crucial insights into disease might we be missing right now by sticking mainly to the coding regions or known gene panels?
That's a great question. What secrets are hiding in the rest of the genome? Something definitely worth thinking about as this technology continues its rapid evolution.